Hello, and welcome to our webinar, Design and Optimize High Multiplexing Digital PCR Assays with the Nika System. My name is Sean McCall, and I will be serving as the moderator for today's presentation. Our first speaker, Dr. Eric Wendlandt, is a field application scientist at IDT. Dr. Wendlandt obtained his PhD in molecular and cellular biology from the University of Iowa, where he studied dysregulation of gene expression in human macrophages upon parasitic infection. As a postdoctoral fellow at the University of Iowa, he investigated how changes in chromosomal structure resulted in drug resistance and disease progression in multiple myeloma. At IDT, Dr. Wendlandt specializes in applications involving gene expression and genotyping, where he helps scientists design and troubleshoot their qPCR experiments on a daily basis. Our second speaker, Dr. Kimberly Gutierrez, is an application scientist manager at Stilla Technologies. She received her PhD in microbiology from the University of Washington, where she studied angiogenesis induced by latent Kaposi's sarcoma herpes virus infection. She completed a postdoc in immunology investigating necroptic programmed cell death and activation of the pro-inflammatory response. She previously held positions at Exicon and QIAGEN as an mRNA application specialist and NGS field application scientist before joining Stilla Technologies. At Stilla, Dr. Gutierrez serves as a scientific liaison supporting the technical needs of the customer to implement Crystal Digital PCR as a multiplexing approach to nucleic acid detection. During and following the webinar, please submit your questions using the Got a Question box. Any questions that are not answered during the Q&A session will be followed up with afterward. I now will turn over the presentation to Dr. Wendlandt.
Thank you for the nice introduction. As Sean mentioned, I'm a field application scientist at IDT, focusing on our qPCR and custom oligo products. A portion of my talk today will be looking at considerations for multiplexing, primarily looking at analysis for compatibility of different assays. The second part of my talk will be looking at some different probe designs for allelic discrimination multiplexing. qPCR assays can be broadly categorized into two types, either primer-onlys, as shown here in the left, or probe-based assays, shown here in the right. As the name suggests, primer-only assays contain two oligos, a forward and a reverse primer. As the primers extend, more and more double-stranded DNA is generated. And an intercalating agent, such as cybergreen, then can bind to this double-stranded DNA. As more double-stranded DNA is generated, the signal increases in intensity. Probe-based assays, on the other hand, contain a third oligo in addition to our forward and reverse primer. This oligo is called a probe. The probe has a fluorophore on the 5' end and a quencher on the 3' end. Generally, the probe produces very little signal until the assay amplifies. Once the extension off of the primer reaches the probe, the 5' to 3' exonuclease activity of TAC begins to hydrolyze the probe, releasing the fluorophore from the quencher. This is then recorded by the instrument as an increase in signal intensity. Each assay type has its own pros and cons. Some of the pros of intercalating dye assays is they're very inexpensive. As you're only purchasing two primers, the cost can be quite low. The longer the amplicon, the stronger the signal. Some of the drawbacks to primer-only or intercalating dye assays is that you cannot multiplex these easily. Non-specific amplification and primer dimers are a concern, and this requires you to run a melt curve analysis after your run is completed. 5' nuclease assays have increased specificity due to the third oligo, which often will add 20 to 30 bases of specificity to an assay. They can be used for SNP genotyping, as well as can be multiplexed by using different dyes on the 5' end. Because the signal is generated from a probe that is specific for your target of interest, there is no need for post-run analysis. The main drawback to 5' nuclease assays is they can be more expensive due to the probe, which is the most expensive component of the assay. For today's talk, we'll be primarily focusing on 5' nuclease assays. I wanted to spend just a few moments discussing some considerations for multiplexing. A few advantages to multiplexing is you can save on reagent and master mix costs. You can also analyze multiple targets in a single reaction. This can help maximize the usage of rare samples, as you only need to input the sample once to assay for multiple targets. Some considerations for multiplexing is master mixes. Not all master mixes are formulated for multiplexing. Oftentimes, they don't contain sufficient polymerase and dNTP concentrations to support multiplexing. So when you begin multiplexing, make sure you are using a master mix that is capable of multiplexing. The Primetime gene expression master mix is one example of a master mix that is compatible with multiplexing. Another consideration is high efficiency assay designs for multiplexing. Each assay should be validated and optimized as individual assays prior to multiplexing. The primers and probes from each assay must be assessed for any interactions, either self-dimers or dimers within the assay or inter-assay dimers. And we'll look at this later on in today's talk. Carefully select dyes for each assay. I recommend that you find the lowest copy number target and label that probe with the brightest fluorophore. And oftentimes this is FAM. Your most high abundant target should be labeled with your dimmest fluorophore. And this is oftentimes dyes in the red end of the spectrum. And the last is lowered plateaus in a multiplex versus singleplex reaction. Each assay is capable of generating a maximum fluorescence output. And when you run these at singleplex reactions, that maximum fluorescent output is oftentimes higher than when you run the same reaction in a multiplex format. And we're going to look at a few examples of this. What's important to remember is that your CQ value should not alter or deviate when you run in a multiplex versus a singleplex assay. For today's talk, we'll be looking at some tools that are provided in the SciTools web suite on the IDT website. They can be found under the tools drop-down menu shown here. The three tools that we'll be looking at are the Primerquest qPCR design tool, the Oligo Analyzer tool, and the Primetime multiplex dye selection tool. The first tool that we will look at today is the Primerquest qPCR design tool, shown here. The Primerquest qPCR design tool can assist with custom primer-only and probe-based assays for PCR, qPCR, digital PCR, as well as Sanger sequencing primers. This can be done by inputting sequences manually or downloading them using GenBank or accession ID numbers. You can either enter sequences individually or using our batch format up to 50 sequences at once using the preset parameters. For more customized assays, our custom design parameters will allow you to specify specific primers or probe sequences, alter amplicon lengths, target exon-exon junctions, and even modulate the TM of those oligos. For today's talk, we're going to be looking at two assays that were designed using the Primerquest qPCR design tool. An assay for human HPRT1, which has been labeled with FAM, as well as an assay for human GUS-B, which is labeled with HEX. Now that we've designed our two assays, we want to make sure that these assays are compatible with each other. To do this, we're going to use the Oligo Analyzer tool found here under Oligo Design and Handling. The Oligo Analyzer tool can provide information on oligo-TM, molecular weight, GC content, extinction coefficients, hairpins, as well as dimer potentials. Today we're going to use the tool to look at dimer potentials between our two different assays. If you're interested in looking at the self-dimer potential, all you would need to do is click on the self-dimer button shown here. But for today's talk, we're going to be looking at heterodimers between our two assays, and that can be found directly below the self-dimer button shown here. In the sequence entry box, I will paste my forward primer from my HPRT1 assay. Click on the heterodimer button, and a secondary input box for secondary sequence will pop up. And this is where I will paste my forward primer from my GUS-B assay. Once I've entered in my secondary sequence, I can go ahead and click on the Calculate button here to look at the dimer potential between these two oligos. Here is the output from our two forward primer dimer checks. What we're looking for here is two things. One, the alignment of the two oligos, as well as the delta G, or how strong that dimer potential is. What I want to try to avoid is having situations like what we see in this first alignment here, having a free 3' hydroxyl. The other thing that I look for is the delta G value. Generally, I want this value to be between 0 and -9. If it falls within that range, it's very unlikely to form the dimer spontaneously within a qPCR reaction. If I do see a configuration like this where I have this free 3' hydroxyl, I want to tighten up those specs a little bit between 0 and -6.5 kilocals per mole. Since this one falls within that range, I'm not too concerned about this dimer. Now that we've looked at the pairwise analysis of our HPRT1 and GUS-B assays, I went ahead and ran the remaining oligos and didn't find any concerning dimers. What I wanted to do is just spend a few minutes looking at some other potential dimers that fall outside of the range that we discussed. Here I have a self-dimer of a primer, which has a delta G value of -15.42 kilocals per mole. This falls outside of the range of the 0 to -9 kilocals per mole that I mentioned. One thing about this dimer that I find interesting is that it's an internal dimer, which means that it's not binding to either the 3' or the 5' end of the oligo. Because this is an internal dimer, it's not of major concern, but if there are other assay potentials or other assay designs that you have, I would look at them first. Try to avoid this if possible. If not, go ahead and move forward with this design. Just keep in mind that these stronger dimers like this may reduce the overall efficiency of your assay. Here is another dimer looking at a primer where once again we have that internal dimer like what we saw in the previous example. This one's not quite as strong at -10.3 kilocals per mole, and it's an internal dimer. What's important though is not just to look at the strongest dimer, but also look at all possible dimers within the output. So since this one's not extendable, it's not a problem, and we can just move on from this and go to the next one in the list. The next dimer in the list isn't going to be quite as strong, and it has a delta G value of -9.3 kilocals per mole. But when we take a closer look at this dimer, it's a little bit more concerning. What we have here is a terminal dimer in which the 3' end will actually bind with itself. In this case, we have a free 3' hydroxyl at the end of one of our primers, and the other primer that dimerizes here actually serves as a template. So what can happen in this scenario is the Taq polymerase could bind and extend off of that 3' hydroxyl, resulting in a primer-dimer extension. These types of primer extension or dimer extension can greatly reduce the overall efficiency of our assay, and I would avoid this type of dimer at all costs. Remember, if this type of dimer were to form, we want to make sure that it falls between 0 and negative 6.5 kilocals per mole. Within that range, it's not likely to form spontaneously within our reaction. The last tool I wanted to discuss today was the Primetime Multiplex dye selection tool that can be found under the qPCR assay design header shown here. The Primetime Multiplex dye selection tool will provide you with a list of dyes that are calibrated for on an instrument that can be used for multiplexing. Here I've selected the Stilla Nika system and asked it to produce three different fluorophore options to multiplex. The recommended dyes are dyes that are calibrated for on that instrument. The alternative dyes are dyes that can be run in a given channel, but may require calibration prior to use. As we'll see in a few minutes, Tim is going to be discussing a multiplex of 6 different assays on the Stilla Nika system. Just because the tool says that it only can multiplex 3 doesn't mean that's the full capabilities of the instrument. These are dyes that are just pre-calibrated for. So if you're looking at multiplexing greater than what the dye selection tool gives you, please reach out to IDT and we can help select the appropriate dyes. Now that we've looked at our assays and ensured that there are no concerning dimers, we have our 2 dyes selected, FAM and HEX. I wanted to spend just a few minutes looking at some data that was generated with these 2 assays. For each assay, we ran them as a singleplex or as a duplex with the FAM hex, using the Primetime gene expression master mix with reference dye and our G-blocks double-stranded DNA gene fragments as our controls. The black lines shown here are the amplification plots when the assays were run as a singleplex. And then the red lines represent what the assay looked like when it was run in a duplex together with HPRT1 and GUS-B. As I mentioned, it's not uncommon to see a decrease in overall fluorescence when running a duplex compared to a singleplex. And that's exactly what we're seeing here. So our singleplex fluorescence in our HPRT1 assay is a little bit higher than what that same assay is when run in duplex. Similarly, we see that same trend with GUS-B. GUS-B singleplex fluorescence is greater than the fluorescence from that same GUS-B assay when multiplexed with the HPRT1 assay. What is important is that our CQ values remain constant, either in that singleplex or multiplex format. And that's exactly what we're seeing is those red lines are overlapping with our black lines for all serial dilutions except for the final dilution. And because this is a very low, this is only 10 copies per reaction, sometimes we do see that, and that's probably more of a setup issue than it is with assay performance. So if you happen to see a little bit shift of the CQ on that lowest dilution, that's probably okay, but just something to make note of. Here's another experiment in which we ran 4 assays as a 4-plex. The top 4 panels are what the assays look like in singleplex format. and then the bottom 4 panels are what those same assays look like in that 4-plex reaction. These were run with the Primetime gene expression master mix again, with varying amounts of cDNA in this instance. As expected, the CQ values remain constant from our singleplex to our 4-plex, but we do see a dip in overall fluorescence. So like I said, it's pretty common to see this, and don't be too concerned if that's what you see in your reactions. With the last few minutes of my talk, I would like to discuss Affinity plus qPCR probes. Affinity plus qPCR probes are IDT-branded locked nucleic acid or LNA probes. And what these are modified bases that go within a probe. And they're actually an RNA base versus a DNA base. How they differ from conventional RNA bases is a secondary bridge that forms between the 2' oxygen and the 4' carbon. What this does is it locks the ribose in an endo configuration. And while in that configuration, it aligns the base more advantageous for base pairing with its adjacent base. This gives us a TM boost of about 2 to 4 degrees. These probes work really well for discrimination because they're very short and they're very rigid, so they don't really tolerate mismatches in DNA bubbling. Because of that, they work really well for single nucleotide changes, SNP detection, as well as indel detection. To demonstrate the power of an Affinity Plus probe, I wanted to show an example from this research paper, Identification of Pre-Leukemic Hematopoietic Stem Cells in Acute Leukemia. In this study, the authors are looking at the presence of a DNMT3A and NPM1 mutation rates during hematopoiesis. What they found in hematopoietic stem cells is there's about a 50-50 mix of wild type and mutants in the DNMT3A SNP. However, the NPM1 is primarily wild type sequence. When we look at the granulocyte phase of hematopoiesis, we see very similar trends with the DNMT3A. It's about a 50-50 mix of wild type to mutant. We do see slightly more of the mutant population than we did in hematopoietic stem cells. However, when we look at the NPM1 population, we're starting to see an increase in the alternate allele or the mutation at this stage. Now that we are looking at the fully matured T cells, we see that in the DNMT3A that the mutant population is essentially gone. Similar, we see that with the NPM1, that there is no mutant population present. Like I said, the biology here isn't really that important for today's discussion, but what's really interesting here is that this study wouldn't be possible without a probe that is able to accurately discriminate even single base changes. And that's one of the strengths of the Affinity Plus probes, to be able to discriminate single base changes within a sequence. That's all I have for today, so I'd like to pass it off to Kim, and she can discuss high multiplexing with the Nika system.
Thank you, Eric. I will continue the theme of multiplexing and how we at Stilla Technologies perform high multiplexing Crystal Digital PCR with the Nika system. At Stilla Technologies, we are dedicated to improving health by offering researchers and clinicians solutions for high-precision genetic analysis with our Crystal Digital PCR platform. PCR is fundamental to genetic analysis and has gone through a technological evolution over time. Traditional PCR was invented in the early 1980s and was the first demonstration of DNA amplification. This first generation of PCR provided a qualitative analysis or simple visualization of a specific DNA target when run on an agarose gel. The second generation of PCR, quantitative PCR, uses fluorescent labeling to quantify the amplification of a DNA target as PCR progresses. Although easy to use, qPCR provides a relative quantification as it requires the establishment of a standard curve with samples of known concentration. These standards may not amplify with the same efficiency as the sample of interest, which may bias the quantification. While qPCR is a widely adopted method for DNA quantification, the third generation of PCR, digital PCR, is a rapidly growing technology. And like qPCR, digital PCR uses fluorescent labeling to visualize target amplification. However, digital PCR does not require a standard curve as it utilizes endpoint analysis rather than real time. And digital PCR also provides absolute quantification of the target and yields increased sensitivity and precision, as we will see in more detail. In digital PCR, the workflow starts with the same reaction mix as for qPCR, containing the reagents necessary for DNA amplification. The next step in digital PCR is the partitioning of the reaction mix into thousands of smaller reactions of the same size. In the Nika system, partitioning is generated by a water and oil emulsion of the mix. As the emulsion is generated, the target sequences are randomly distributed in the droplets, such that some droplets contain one or at least one sequence of interest, and some don't contain any sequence of interest. Each of these droplets now acts as an independent reactor where PCR can occur. A successful PCR will lead to the generation of a fluorescent signal at the end of the reaction within the droplets that contain the target of interest. The fluorescence intensity of each droplet is then plotted via a dot plot with two clusters separated by a threshold automatically set by the analysis software. The droplets above this threshold are considered positive, while the droplets below the threshold are considered negative. The analysis software then uses the proportion of positive droplets and Poisson statistics to estimate the concentration of the target of interest together with the uncertainty on the measurement. The latter lies in the fact that a positive droplet may contain more than one target sequence and is dependent on both the concentration of the target in the PCR mix and the number of droplets generated. Crystal Digital PCR with the Nika system applies a patented partitioning technology to create a self-assembled array of droplets called a droplet crystal. In a droplet crystal, the size of the droplets is fixed and controlled via the microfluidic design. Ensuring a monodispersed population of droplet size is important in order to guarantee a random distribution of the target sequences within the droplet crystals. Another feature of the Nika system are the 3 or 6 fluorescent channels, allowing for the quantification of multiple targets in a unique sample. In this example, 2 variant sequences of genomic DNA can be quantified together with the wild-type sequence. In such an example, the lowest abundant target, represented here in red, is artificially enriched by partitioning, making it less prone to competition, as would be the case in a bulk reaction in qPCR. This is why digital PCR is more sensitive than qPCR to detect rare events. The Nika system workflow consists of 4 steps: prepare, amplify, read, and analyze. Collectively, the Nika system provides a complete, easy-to-use, and fully integrated system to perform Crystal Digital PCR. We will explore each of these steps in more detail. Beginning with the preparation of your digital PCR reaction, we have recently launched our line of Nika PCR mixes optimized for enhanced compatibility with Crystal Digital PCR and performance on the Nika system. Simply choose the PCR mix that matches the fluorescent chemistry of your assay, either probe-based or dye. All mixes contain a hot start enzyme and are available in 5x or 10x concentrations. The 10x concentration is ideal for high multiplexing needs and allows for more sample volume in the reaction mix, thereby increasing the detection sensitivity. The heart of our innovation is the Sapphire Chip, which integrates the droplet crystal technology. The chip is pre-filled with oil, thus the only manual step is to load 25 microliters of PCR mix in each of the 4 wells. A droplet crystal with up to 30,000 droplets is created by contactless fluid injection. Due to the microfluidic design of the chip, each droplet has a fixed volume of 0.59 nanoliters. Given these specifications, the dynamic range of the Nika system spans 5 logs with an LOD at 95% confidence of 0.2 copies per microliter up to 20,000 copies per microliter. To load the chips with PCR mix, you simply have to open the inlet port, pipette 25 microliters of mix on top of the oil already inside, and seal the inlet port with the white vented cap. Once loaded and sealed, up to 3 Sapphire chips, or 12 samples, can be placed in the first instrument, the geode. This instrument will perform both the partitioning of the sample as well as target amplification by following the PCR cycling conditions defined by the user. At the end of the PCR reaction, simply transfer the chips from the geode to the Prism, which will read the endpoint fluorescent signal of each crystal in up to 6 channels. The probes listed here are recommendations for each channel. However, custom probe combinations can be selected as long as they fit within the wavelengths displayed here. After scanning on the Prism, data visualization and analysis is performed with our Crystal Miner software. This software is developed completely in-house and allows a guided and intuitive analysis of the data for an in-depth understanding of the results, as well as a robust and reliable quality control. Crystal Miner is available with no limitations on users or licensing fees and can be downloaded together with demo data from our website. The Nika system provides a complete solution enabling users to perform Crystal Digital PCR in under 3 hours with minimum hands-on time. The integration of the workflow within the Sapphire chip makes it robust against contamination and very easy to use. And our Crystal Miner software is an open and intuitive program giving you complete control over your data and analysis. The Nika system is compatible with many sample types to address a wide range of applications. Advantages of this easy-to-use digital PCR workflow will benefit your analysis for absolute quantification of DNA or RNA, copy number variation assessment, rare event detection, or gene expression analysis. The Nika system is uniquely designed for multiplexing assays aimed to maximize the data output from precious samples. However, as discussed by Eric in IDT's presentation, creating successful multiplex assays requires some level of optimization. A real advantage of Crystal Digital PCR is the ability to visualize suboptimal assay interactions in greater detail than in qPCR. For example, the presence of false positives can easily be identified in the 1D dot plots of a no template control. Accurate and proper controls are important in order to interpret your results correctly, and false positives can negatively impact the sensitivity of your digital PCR assay. The presence of rain or partitions of intermediate fluorescence suggests that amplification or hybridization efficiency is suboptimal. The presence of rain makes setting the threshold more difficult and thus may affect quantification. Poor separability between negative and positive populations also renders thresholding and accurate quantification difficult. And finally, the presence of unexpected populations is an indication of specificity issues within the assay. Taking the time to carefully design and optimize your multiplex assays will greatly impact the accuracy of your Crystal Digital PCR results. The Prism is equipped to detect targets in 6 distinct channels, thereby supporting high multiplex assays. Here I'm showing the 6-plex validation assay for the Prism designed by Stilla. This assay combines 6 unique targets run simultaneously in a single well. As you can see in the 1D dot plots on the right, each assay exhibits clear separability between the positive and negative populations, and very little rain is visible, allowing for easy and accurate thresholding to be set. Given our ability to multiplex, we can help provide support to our clients to design and optimize custom assays for the Nika system. I'll highlight one such example on the next slide. We collaborated with JBS Science to design and optimize an assay to detect wild type and a single nucleotide polymorphism located in a mutational hotspot of beta-catenin from liquid biopsy samples. JBS Science specializes in isolating circulating cell-free DNA from urine with the aim to improve cancer screening. These samples require increased PCR sensitivity to detect the low levels of mutations present. Therefore, Crystal Digital PCR was implemented in the workflow. Urine cell-free DNA is highly fragmented, so the first challenge was to design a small amplicon of less than 60 base pairs to cover this mutational hotspot. Next, we had to take this small amplicon and create a duplex assay with probes to distinguish between the wild type and the SNP. Therefore, for accurate and precise quantification, the probes need to be highly specific. Stilla provided the technical support and utilized the IDT design tools to create the duplex assay based on the parameters outlined by JBS Science. And JBS Science provided the DNA extracted from urine for analysis. With Crystal Digital PCR, we were able to quantify both the wild type and the SNP in a single assay. To demonstrate the specificity of the assay, we performed a serial dilution of mutant plasmid spiked into a wild type background. We were able to detect the mutant down to 0.41 copies per microliter in the presence of the wild type. We're continuing to work with JBS Science to expand this design to cover more mutations located within this hotspot of beta-catenin. And while this is an example of a duplex assay, I'll now talk about a 6-color panel currently being used in a clinical study. As part of the Horizon 2020 Lima project, we have developed a 6-color panel to monitor breast cancer mutations. This project is part of a clinical study combining liquid biopsy monitoring and MRI scans to predict cancer therapy responses. 100 patients are enrolled in the study, with 10 samples per patient monitored by this panel with Crystal Digital PCR. This project highlights the reliable, rapid, and cost-effective evaluation of breast cancer mutations covering 25 to 35% of patients from a single blood sample. This 6-color breast cancer panel is designed to monitor 3 PIK3CA mutations, HER2 amplification, SS chromosome 17 polysomy. An exogenous positive control is also included to monitor for PCR efficiency. A limit of detection of 0.8 copies per microliter was established for PIK3CA screening. Preliminary validations of the assays were performed on patient samples, and the experimental results tracked closely with the theoretical presence or ratio from each patient. The number of unique data points obtained simultaneously from one sample underscores the power of multiplexing with Crystal Digital PCR. And this study is currently ongoing in the Netherlands. In addition to our 6-color Prism, which we'll be launching later this year, Stilla is developing a number of 3- and 6-color kits for the Nika system. These include an already available 3-color kit to quantify SARS-CoV-2, and coming soon, a 6-color kit to detect EGFR mutations associated with lung cancer. If you would like to experience Crystal Digital PCR in person, or would like your samples tested on the Nika system, Stilla has a demo lab and training center located just north of Boston. Digital PCR principles, hands-on demonstrations of the Nika system, and software trainings are all conducted by our Stilla-trained application scientists. please reach out to us for more information about an on-site demo. I'd like to thank you for your time, and a special thank you to JBS Science and Selena Lin for our continued collaboration developing assays to detect mutations from cell-free urine on the Nika system. For more information about Stilla, our products or workflows, please visit our website at stillatechnologies.com.
Thank you, Dr. Wendlandt and Dr. Gutierrez, for your very informative presentations today. We have received several questions from our audience. The first question I have is for Dr. Wendlandt. What do I do if my instrument is not listed on the multiplex dye selection tool?
That's a good question. We try to update that tool on a regular basis and keep as many of the newer instruments listed on there. If you do run across an instrument that maybe is a little bit older or just released and it's not on the tool, please feel free to reach out to the application support group or myself, and we would be happy to help you select the appropriate dyes for your instrument.
Okay, thank you. The next question I have is for Dr. Gutierrez. Can you use any master mix with the Nika system?
Yeah, we actually recommend that you use our Stilla branded Nika master mixes because these have been specially designed and created and optimized for use with Crystal Digital PCR. There are other mixes you can use, but we would have a really hard time supporting you technically if there were any issues with the droplet formation or the assays in particular.
Okay, excellent. Thank you. The next question I have is for Dr. Wendlandt again. I noticed on the website lists only a few different dye options. Are those dyes all that you offer for qPCR?
We have quite a few different dye options available. The website just starts to scratch the surface on what's available. If there's something that you're interested in and it's not listed on the website, feel free to reach out. We are able to accommodate virtually all different dye quencher combinations, and it just might be something that we have to process via email and not through the web accounts.
Okay, thank you. Dr. Gutierrez, for the 6-color Prism, are you limited to the 6 fluorophores shown in the presentation?
No, absolutely not. As you can see, we give the wavelength, the excitation, and the emission spectrum for each channel. So as long as your fluorophore fits within that spectrum, then you should be able to know which channel that fluorophore will emit in.
Okay, thank you. Dr. Wendlandt, are there other options for allelic discrimination assays other than the Affinity Plus probes?
Yeah, there is. Affinity Plus is our go-to for allelic discrimination just due to the customization and the flexibility of the probes. We're able to offer an MGB probe through our GMP lines and soon as an RUO product. So MGB probes are also available. There are some other technologies that are available, but they're not going to be compatible with multiplexing.
Okay, thank you. Dr. Gutierrez, does Stilla have any off-the-shelf assays?
We do have a few oncology-focused 3-color assays that have been created with a partnership that we have with ID Solutions. As I said, we also have the SARS-CoV-2 kits, and we will be coming out with some 6-color kits along with the launch of the new Prism 6 as well.
Okay, thank you. Dr. Wendlandt, question here. I noticed in a multiplex reaction I ran that the CT values differed from the CT values of my singleplex reaction. I didn't notice any dimers, so what could be the cause of this?
So when we run the dimer checks like what we discussed in the presentation today, we're doing a pairwise analysis. as we get into more and more complex multiplexes, there's numerous interactions that can occur. And it's not necessarily just a pairwise analysis. So what could be going on is that there are predicted dimers, there's dimers that are not predicted to form that are actually forming. So this could be due to the high level of nucleic acid within the reaction. It also could be due to a multi-configuration, so maybe multiple oligos, more than two are coming together to form these dimers. There are ways to help relax these dimers and restore some of that efficiency. So if you do run into situations like this, feel free to reach out, and I'd be happy to direct you to kind of workarounds for those scenarios.
Okay, excellent. Thank you. Dr. Gutierrez, how many targets can you detect with the 6-color prism?
That's a great question. Just because the new prism has 6 different channels does not mean that you are limited to detecting just 6 targets. In fact, our new EGFR kit that we are developing actually detects 10 different targets using those 6 channels. So it is possible to detect multiple targets even with a limitation of 6 channels.
Okay, thank you. And then Dr. Gutierrez again, can I send samples to Stilla to be tested on the Nika system?
Yes, absolutely. We do have a demo lab and training center, and we would love to test your samples, and we would provide you with a sample report and go through all of the data with you and explain everything with one of our application scientists.
Excellent. Wonderful. Thank you. Unfortunately, we have run out of time to answer additional questions. We will contact you directly if you asked a question and it was not answered here today. Again, I would like to thank Dr. Wendlandt and Dr. Gutierrez for their time with us today. Lastly, thank you again for attending, and we wish you the best of success in your research.